So when we're trying to figure out in the brain, like, what is connected to what? Like if you look at a particular nucleus, for example, like what is bringing axons into that nucleus, right, to form synaptic connections with the dendrites, and cell bodies in that nucleus. And then also you want to know, where does that nucleus then project to? You want to trace all the highways and byways in the brain. And to do that, scientists have made use of these chemicals that are called tracer chemicals, tracers. And tracers can be what we call anterograde, which means they get picked up by, the neuronal cell bodies or the dendrites at the site of injection where you put the chemical in. And then they get transported down to the axon terminals and collect there. So then you can sort of visualize them. And then there's also what we call retrograde tracers, right? Which are picked up, let's say, if you put it into a nucleus here, they'd be picked up by the axon terminals here and then get transported back, and they can stain, whatever structure was transmitting, sending information into that particular nucleus. So the retrograde, tracers take you back. So you can have monosynaptically restricted anterograde tracers. That means like only one synapse. So basically, like if you were to inject in this site, it would only go to the ends here, right, and stain the ends somewhere else. It would just go one neuron, right? But you could also have what are called polysynaptically linked tracers, which would, basically if this is the nucleus right here, you inject it, gets taken up by the cell body here, transported down to the axon terminal, and then passed into another neuron, and then works its way down to the end there. So you could sort of see like a chain of interconnected structures utilizing these polysynaptically tracers, polysynaptic tracers, anterograde tracers. And if we're talking about retrograde, it goes the opposite direction. It means it'll get picked up by the axon terminals, go back to the cell body here and the dendrites, and then it may even stain further back into another structure kind of beyond. So tracers are categorized as anterograde, that's the forward direction, retrograde, that's going back to where the projections are coming from, based on their direction of travel within neurons. And there are a whole bunch of now what we call fluorescent tracers. There's one like fluororuby or fluorogold, for example. There's a whole bunch of really brilliantly red and brilliantly green ones. These are anterograde tracers that can be injected into a structure and either go forward in the anterograde direction, be carried down to the axon terminals and stain, where a region where you've injected is projecting to via those axonal connections, or it could be picked up and transported in a retrograde direction. So in many papers, you'll see really beautiful, kind of images of brain slices with these fluorescing, structures. And I've got some examples on our website here of what that looks like. So another type of tracer is something called a metabolic tracer. And this is something that collects in areas of greater metabolic activity. So one example would be like if you radio label glucose, for example, and areas of the brain that are being more active, under certain task conditions, for example, are going to take up more of that radio labeled glucose and incorporate it, into their ATP and into their, the structures that, into cellular respiration. So you're going to get a greater signal, basically, from areas that were active, during a specific task that had higher metabolic activity. So that's another kind of form of tracing, that can be done using chemicals to figure out functionally, in that case, what areas are active under certain task conditions.